Good evening, folks, and welcome to the 35th Leonard A. Ford Lectureship. I'm Dr. Jeff Pribble, and on behalf of the Department of Biochemistry, Chemistry, and Geology and Minnesota State University Mankato, I welcome you to the 35th Leonard A. Ford Lectureship. Who is Leonard Ford? We have slides up behind me. Dr. Leonard Ford was a professor of chemistry at Mankato State College from 1939 until his death in 1967. He was chairman of the chemistry department from 1939 to 1947, chairman of the Division of Sciences and Mathematics from 1947 to 1965, and coordinator of special programs and chemistry professor from 1965 until his death in 1967. Dr. Ford made significant contributions to science education during his time at Minnesota State Mankato. He published articles in journals such as Science Education, School Science and Mathematics, and the Journal of Chemical Education. He also published articles with the American Chemical Society, the Minnesota Academy of Sciences. His book, Chemical Magic, was first published in 1959 in hardback and later in 2 editions in paperback. This book also achieved international readership, being published in French, Spanish, Danish, and Japanese. Leonard Ford established the science fair at Minnesota State Mankato in 1951, and that science fair continues to this day. Dr. Ford was instrumental in establishing the nursing program, medical technology program, dental assistant program, dental hygiene program, and engineering programs at Minnesota State Mankato. Dr. Ford was recognized by his professional community as a leader. He was president of the Minnesota Academy of Sciences. He served on the Education Committee of the American Chemical Society and was committee chairman, science section of the Minnesota Education Association. He was also president of the Mankato Lions Club and served on the Board of Trustees and Alumni Board at Gustavus Adolphus College. This lectureship receives generous financial contributions from the Ford family, especially Dr. Ford, Barbara Ford Olson, the daughter of Leonard Ford. This is the first time in many years that Dr. Olson has not been able to join us, and we hope to see her back here next year. With us this evening is Eric Ford Olson, the son of Dr. Barbara Ford Olson and the grandson of Leonard A. Ford. He would like to share a few words with us, so welcome, Eric.
Check. Thank you, Jeff. I just really want to honor the legacy of my grandfather. I'm really glad for me to be. It's been many years since I've been at this particular the lecture here, and I just on the way over here today, I went over to the graveyard where my grandfather and grandmother are buried, and I had a little moment with them in their final resting place, and he died when I was just 1 year old, so I really never got to know him, and got to know him more from his legacy. And one thing that's really interesting for me is that he was very much a scientist, left brain, analytical thinker, but he was also a musician. And he was the one interesting story is that he was in the Depression. It was hard for him to find an academic teaching job, so he actually got a job at a military boys' school where he taught chemistry. But then there was a band leader that had a really difficult time managing all the kids in the band were misbehaving, and so my grandfather, in addition to his teaching chemistry, he taught the band, and he could play all the instruments in the band. Apparently, he could play all the instruments, and in addition to that, he played piano and violin. And myself, I am a civil engineer, and so I get a lot of my left brain from him. I think my mother and myself, and then also music, because I'm a multi-instrumentalist musician myself. I play many many instruments. And so I really think it's a beautiful combination. I don't know how many of you out there might be musicians, but it's a powerful combination to be very much into science, but also very much into music. So I encourage you all to take up creative expressive paths in addition to the scientific paths. And I just want to honor him by when I'm being scientific and when I'm being musical, because I feel like I get that from him. So thank you all for attending tonight, and looking forward to our speaker tonight. Thank you.
Thank you, Eric, for those memories of your father and your grandfather, things I did not know. Our lecturer today is Dr. Nick Riley, assistant professor in the Department of Chemistry at the University of Washington. Dr. Riley earned his BS degree in chemistry from the University of South Carolina. Then he moved to the University of Wisconsin-Madison, where he earned his PhD studying with Professor Josh Kuhn. He completed a postdoc with Professor Carolyn Berzozzi at Stanford University. And in his current position, his research group uses state-of-the-art mass spectrometry and chemical glycobiology to investigate glycoproteometric regulations. Told you I would mess up some of those words. This year, Dr. Riley was named as one of the chemical and engineering newses from the American Chemical Society Talented 12. CNE News asks readers to nominate early career scientists who use their chemistry know-how to make a real-world impact. Dr. Riley rose to the top of almost 370 individuals nominated. In a recent CNE News article on the Talented 12, Dr. Riley states that his lab's superpower is, and I quote, "I would like to think that it is my data analysis and figure-making skills. But really, it is that I can reach things on high shelves because I am tall." Unquote. Please join me in welcoming Dr. Riley to Minnesota State University, Mankato.
Evening. Test to make sure the mic works. I think I can hear it on, so I think we're good. It's wonderful to be in front of a few familiar faces again from this morning's presentation, so hello again, and hello to the folks who I haven't interacted with yet. It's a real pleasure and honor to be here in the name and honor of Leonard Ford, and it's a very cool legacy to now be associated with, and to even hear stories about his interactions with people in the state of Minnesota, his ideas for science and music together. It's a cool space to be a part of, so I really much appreciate the department here and your university for hosting me. I see, because I know familiar faces in the audience, I want to say the first intro slides here are going to look very familiar, but then we're going to dive into something that we didn't talk about this morning. But the way I started this morning was asking for a show of hands, how many people have been to Seattle before? And I see that show of hands too. Okay, a fair number of people in the audience. And if you were here this morning, you'll know that looking at a picture of campus. Make sure this is up that way. And so this is my walk into campus. So this is a picture from my phone. And just to the right of that fountain is our chemistry building. So I get to walk down this and see this often. This is from April when we have our cherry blossoms in full bloom. And if anyone was here this morning, you want to help the audience out, there's something that's here that we can't quite see. Does anyone remember, if you were here this morning, what that would be? I hear whispers. There's a giant mountain that you can't see. And I like to use these images precisely because you can't see it during these conditions. But if you look in a different way, at a different time, in a completely different setting, it becomes very clear, hopefully with the lighting it's there, it's a dramatic feature of this viewpoint. And you totally miss it if you're not looking in the right way or at the right time. In this, we talked this morning about how it's about analytical tools that allow us to look this way. But I also want to use this as a pivot point to thinking about these things we're going to talk about today called glycoproteins, and we're going to get into all what those words mean. But in biology, glycoproteins are often kind of the forgotten child that have become a bit more in vogue in recent years, but often we just totally overlook them even though they're staring us in the face. And so this is also a good metaphor for what we think about when we talk about these molecules we're studying today. And another thing I wanted to point out, I didn't show this morning, but as a quick reference, if you haven't been to Seattle, that there's 3 national parks that surround us. So there's mountains everywhere, there's water, so definitely please come visit us and check this out. But I also wanted to point out like how far away Mount Rainier is. So that mountain we were just looking at, that's a huge part of that image, is a good 80 miles from where we were just standing in that picture. And also you can see here just mountains everywhere. So it's just, again, to say a cool landscape and a dominant feature that's that big from 80 miles away that we totally miss. Also, because I get to walk in this way, if you were here this morning, you know I have a lot of pictures from that vantage point. So the next one I'll show, one of my favorite that I've taken, was from one morning a few months ago. And I use this one because it is a beautiful area, and what an impressive geography that it has. And as the University of Washington, we are occupying these lands that were originally occupied by Native peoples. And so we have this land acknowledgement statement to make sure that we cover and acknowledge the fact of where we have built our university and where we study our science. And one last point that I have is my friends often will say, well, you show pictures of Seattle when it's beautiful. I hear about this rain, so I've selectively cherry-picked, again, pictures from my camera roll of all 4 seasons, just to show you that if you do come, maybe there's a chance you see the mountain. And we actually have a full social media account that says, is the mountain out? And it just posts yes or no with a picture. So you can at least check before you visit. And then finally, the last picture of this is from a sun setting. And this is a quote I use recently, in recent months, as really a statement of solidarity. We know across federal funding in science and for higher education, it's a struggle at the moment. And so this is just a statement to say for people who want to keep investing in higher education and in science and in the progress towards this world of biotechnology that we're interested in, to keep fighting the good fight and to keep pushing for progress in that way. And the reason I think it resonates well with the Ford Lectureship is to think the reason to keep pushing for that progress is because of trainees and because of scientists that will be the next generation. And so I think a lot about what Leonard Ford did for bringing science to people who weren't always first and foremost scientists, and also thinking about how to bring the next generation into the forefront of what we can do with the tools that we have at our disposal. So this is my research group, and today we're not going to really talk much about research per se, but more introducing cool concepts that have led us to kind of the type of research that we work on. So my research group there, and we'll end with this slide as well. But okay, the last thing that's going to be similar from this morning is this slide, and then we're pivoting away, I promise. But I wanted to just give a brief overview of kind of where I've been. And you heard kind of about this with the introduction, but I'm originally from Kentucky, went to undergraduate at South Carolina. There I had several different majors. We were actually talking about this today at dinner, where I was interested in chemistry and psychology and criminal justice. I was thinking about a lot of different things. I ended up choosing research, but then what I would like to highlight about this is I get to then travel all across the country exploring this passion. And so it's a cool world to be in, to think that science can not only broaden what you're thinking about, but also the spaces you get to interact with, the communities you get to build. So from South Carolina, I moved to Wisconsin. I was woefully unprepared for winter when I moved there. Had to buy an entirely new wardrobe for that. I really enjoyed my time in Madison working on mass spectrometry. And so this morning we talked a little bit about what mass spectrometry is, how it can be useful as a tool to study these molecules we're going to talk about. So today we're not going to focus so much on that. We're going to focus more on what I thought about during my postdoctoral studies in the Bay Area. So I was at Stanford University with Carolyn Bertozzi, and she was the 2022 Nobel laureate for developing chemistries to study the exact biomolecules that we're going to talk about today. So a lot of my learning and inspiration for this comes from her and the teachings that I learned in her lab. And then I just moved to Seattle 2 years ago to start my own group there. So you kind of see my path in decided to stay on the West Coast for a few years here. So okay, now is where we kind of pivot away from where we started this morning. And hopefully, if you haven't gotten a bag of M&Ms, I know they're floating around here somewhere, but the reason M&Ms were paired with this talk is because it directly relates to ideas I want to talk about. And believe it or not, your cells are coated with sugar, just like an M&M. And so that relates back to the title of this talk, how the cell's sugar coating can inform interesting things about their biology. The difference is, when you think about sugar often, you're thinking about the sugar you eat, carbohydrates that would be either sweetened things or in bread or whatever it might be. And so in this case, that sugar is not the same sugar we're talking about. We're going to kind of shift a frame of reference. So by the end of this talk, you'll be like, I know what cool kind of sugars are on our cells, and hopefully gives you a frame of reference for thinking about these molecules you may not have interacted with much before. And so one thing I use to kind of introduce this world to is that the sugar coating on M&Ms is static, right? So that doesn't really change until you eat it. But with the cells that we're going to talk about, that sugar coating is changing as the biological system changes, and that gives it a really cool set of properties that allows us to understand how cells interact with their environment. But before we dive into that, I want to start kind of at the basics and build up to some key words that matter to this conversation. And so first, I think most people in this room will likely be familiar with the idea of the periodic table. We have all of these elements. And so these elements are going to be the building block of where we're going. So if you haven't looked at this in a while, just to refresh yourself that we have elements, and in the biomolecules we study, we have ones that are pretty common, like carbon, nitrogen, hydrogen, oxygen, sometimes sulfur, phosphorus, things like that. So most of the biomolecules we're thinking about are in that space in the periodic table. When we then think about what a molecule is, right, it's a combination of different atoms from these elements that are then representing the fundamental unit of a reactive species. And so these would be molecules that we interact with in our daily lives, caffeine being probably the most important of all that are on the slide. When I say molecules, we're thinking about collections of atoms that are arranged, right, in chemically important orientations. From there, that's where the word biomolecule comes in. And so these are the molecules of life. And there are different classes of biomolecules that you may have heard of before, but in case not, wanted to make sure that we covered this base. That we have things like lipids, which are fats, fatty acids, maybe words you've heard before. Nucleic acids would be our DNA, our RNA. Carbohydrates, so those are the sugars that we're talking about this evening. And then proteins, which we'll also talk about too. So when we think about this world, I also want you to think about how these are organized into the bigger picture organisms and systems that we think about kind of at the macro scale that we interact with. And so maybe not at the macro scale, but at the micro scale, we have things like organelles. So we have proteins, lipids, sugars, RNA and DNA that are packaged into small pieces that then are packaged into what we call the fundamental unit of life, which would be a cell. And so within that, we're going to kind of briefly touch on this, but we're going to really stay in this space for most of today. But of course, if you're not used to thinking at the cellular level, the cells are then built into tissues, tissues into organs, organs into organ systems, which would then make organisms. So this is kind of the space we're thinking about. We're really going to be thinking at this level for most of our conversation tonight. This is a great artist rendition, speaking of the combination of art and science. If you haven't heard of David Goodsell before, I highly encourage you to write down his name right now and go look up his artwork. It's phenomenal. He uses structural biology to understand what are the shapes and orientations and structures of different biomolecules within living systems, and then he hand draws them in really cool representations of life. This is a representation of a cell that he's drawn. And here we can see that we have DNA and RNA that are shown by the kind of yellow pieces here. We have proteins that are in the cyan and the pink. And then we have some actin proteins in the yellow, like stringy looking stuff. And then here we have a lipid membrane. And this is when we think of cells, they're encapsulated in this lipid membrane. And what we're thinking about today is what's on the surface, these sugar molecules that might interact. You can kind of see how this could really affect how cells interact with their environment. So with that, let's think a bit about how we get to all of this composition of biomolecules within a cell. How do we get to where sugars are going to play an important role here? So first, DNA, probably the most common of any of the biomolecules folks have interacted with before. So these are nucleic acids, and these are really the blueprint of life, right? This is the instruction manual for any given cell that tells how to make all the different biomolecules that you might need. And some words that I'll use, and I don't want to skip over them without defining them, so I'll add a few definitions as we go. And so the idea of a genome would be all of the DNA that is in a cell or a system that we're talking about. And so this would be, the genome would be all DNA and all the genes that are there. And to think about what the genomic era was, or the era of the genome, really came to fruition in the 1990s and 2000s. And this was when all sorts of technology advances allowed us to sequence genes much faster and cheaper than we ever had the ability to do before. The technology had existed for multiple decades, but the ability to do it at a scale where we could do entire organism sequencing was really the revolution of this time. And so when we think about what scientists were working on in this era, they were trying to say, what are the genomes of different organisms? We have eukaryotic systems, like yeast would be one of the most simple of these systems. And it came out in the mid-90s or so that the yeast genome is about 6,000 genes. And the idea was that the more complicated the organism, the more genes must be there to explain this biological complexity. So as we were exploring the genomes of all of these different organisms, we started seeing things like C. elegans, which are nematodes, little worms. They had 15,000 genes. That's more than 6,000. That kind of tracks with our expectations. A big one would be the fly genome. A lot of work, really great genomic work done in fly systems. Flies have 20,000 genes. Okay, this again is tracking. And so then at this era, you know that the human genome was released in 2000, in the year 2000. And so the expectation, some people were saying, are there 100,000 genes in the human genome to explain this leap from biological complexity from a fly that has 20,000 to humans that have however many? It turns out one of the great surprises of the Human Genome Project is that humans only have about 25,000 genes. That's not that much more than a fly. That's not that much more than a nematode. That's not that much more than even yeast. So the question became, how do we get biological complexity that we know as humans and other higher organisms from a similar genome size? So this is where things get interesting. If we expand past just what's the blueprint, what's the DNA, we have this central dogma of molecular biology. And this tells us that the DNA blueprint gets read into RNA, which then gets, we call that process transcription, and then the RNA gets translated into proteins. And the reason for this, right, you wouldn't want to take a blueprint, and if you messed it up or somehow damaged that blueprint, your plan is forever ruined. So what we do is we basically copy that plan into a carbon copy that is that RNA, and that then becomes our template to make proteins. But if the RNA gets damaged or gets modified in some way, we still have our original blueprint that's there. But then what's interesting is that translation to protein is where we start adding some different layers of functionality. Words that you'll hear in this kind of context, again, we talked about genome. We have the transcriptome, which would be all of the RNA molecules that have been transcribed from DNA. And then the proteome is the collection of all these proteins that are there. And so then how do we get to the idea that we have this blueprint of what to make, but now we're talking about molecules that we refer to as effector molecules because they affect things in cells. They are the doers. And proteins and things that are downstream of this process are what we call the effector molecules. So they're the people that are if we had to analogize this into humans, it'd be the people building the things or doing the actions that we're foretold in the blueprints. And so what grew out of this idea of how do we have so much biological complexity with a relatively, quote unquote, simple genome, comes to do with this idea of post-translational modifications. And if you remember, translation is when we made proteins. And so here, the human gene that we might have, this is just a single gene we're looking at that's DNA, can be made and transcribed into one RNA, maybe even multiple different versions of RNA. Those can then be translated into protein sequences. And now we have proteins that should be doing functions within our cell, but we then can modify those proteins even further. And this layer of regulation is where that biological diversity comes from. And this is what it becomes a really interesting part about studying biomolecules in living systems. And so at the end of the day, what we are interested in studying are these collections of molecules called proteoforms. Proteoforms would be the versions of proteins that exist with modifications or without. It's the collection of all of the proteins that were maybe encoded by the genome, but then also have some bonus modification on top of that. And what we're focusing on today is this one called glycosylation. We're going to dive into this one in particular, and it is arguably the most complex, and I would say the most interesting of this set of post-translational modifications. And so before we dive into that, why do people care about studying the proteome? Well, it goes back to this idea of the plan versus the action. And studying the genome or the transcriptome can tell you the plan, but we need tools to be able to study proteins and then small molecules like lipids and sugars. And I would argue, hopefully by the end of this you would believe me too, that it's not just the proteome, but glycoproteome, things that are proteins that have these sugar molecules attached that we want to study. So let's go back to then this argument and this view of what a cell might be. And so now we understand that the M&M is representing this unit of biological information. We have the sugar coating on top. And if we were to translate this into an image of an actual cell, these are the types of images that we can take. And so what we're doing here is this would be a bacterium cell. This is from a mouse myocardial epithelial, a capillary, so this is a mammalian cell. And we've stained for negative charges. And so here, all of the fuzz that you see here are negatively charged glycoconjugates. And so this word that we'll keep seeing, glyco, I'll just go ahead and skip to this, that prefix is the Greek prefix for sugar. So when you see glyco, think sugar. And what this collection of all of these glycoconjugates, i.e. things modified by sugars, we call that collection the glycocalyx. And that calyx word is also the Greek root for husk. So sugary husk is what we're thinking of in this context. And so our question would be, is that fuzz look the same all of the time? How does that change as a cell changes, as a healthy system moves to a diseased state, or any of these state changes that we might want to understand and study? I'll also add that black and white is just how we made this measurement. As an analytical chemist, I have to point out the measurement science behind it, and that you don't have to image in this way. If you image with a completely different method, that you use something called a fluorophore that sticks a color onto these molecules, you can also get interesting, in this case, red staining of the sugar molecules. Just wanted to point that out. Now, when you think of what this glycocalyx represents, or like how to maybe conceptualize this in a different frame of reference, a way that's useful is to think about vegetation on the surface of the earth. We go back to even the mountainous world of Seattle and thinking about all these forests that cover it. The glycocalyx is not so different than that, in that it's really a complex set of decorations. Again, vegetation decorating your cell is just an analogy here, but this would be a model of just a single glycoprotein where this red is the protein and the green and yellow are all the sugars that are decorating it. So you can see how maybe they represent the trees or the forest on top of our cells. What is the forest made of? Well, we start with this idea of monosaccharides that then get built into larger sugars called glycans. And so this is where I want to make sure I have these definitions down, where the glycome, instead of the genome, the transcriptome, the proteome, we now have the glycome. And this is the collection of all of the sugar molecules in the system we want to study too. And now, sugar molecules, we have building blocks we want to think of. We didn't go into building blocks of proteins or amino acid, of nucleic acids, things like that, but those also have building blocks. And similar to that, sugars have things like sialic acid would be one, glucose, galactose, mannose, and so all of these have structure. And as you see here, we kind of represent these with different shapes and colors. And these shapes and colors are represented in these light-colored boxes as well. And it's easier to use these shapes and colors because GLICNAC, or N-acetylglucosamine, and GalNAc, or N-acetylgalactosamine, are isomers. And the only difference is the position of that OH group, equatorial axial. And so having to look at that in a collection of sugars would get a little dense, to say the least. So we represent glycans using what we call SNFG, or symbol nomenclature for glycans. So you'll see what we call these Lucky Charm type figures representing these sugar molecules. But to know that they have chemical structure, and this chemical structure is what imparts their function. So now we know if you hear the word glycan, it's the idea of these monosaccharides put together in some combination. And this combination and the structure here all matters. So when we were to try and draw the glycocalyx, these trees, this vegetation on our cell surface, we can draw them using all the different types of glycans that might exist in the glycome. And so this would be our plasma membrane right here. So anything below this would be intracellular, but all of this space is extracellular. So now the sugar is decorating the extracellular space. And what I want to impart upon you here is how heterogeneous this is. I'll first point out that we have lipids here. So lipids can be modified by sugars. Call them glycosphingolipids. We have proteins, and we call these N-linked proteins because these types of glycans are linked through a nitrogen on a protein side chain. And these N-linked glycans have specific structures and combinations of monosaccharides that differ from something like an O-linked glycan. So here the N and the O represent how it's attached to a protein. And even look at the difference in what an N-linked versus an O-linked glycan structure looks like. It's not just that the monosaccharides are different, but how they're put together also changes. From there was all these other classes where we have different modifications, whether it's glucose or GLICNAC or mannose. And so there's this heterogeneity that dominates. That's like the main feature of glycosylation at the cell surface. And so we're going to talk about why cells might use so many different types of biomolecules for these purposes. But again, I point out that these are representing actual monosaccharides and chemical structures. So if we were to put this together, this would be an N-linked glycan. This is what the structure looks like. And why I wanted to show this, we showed this morning as well, is to maybe give you an idea. We don't have any dynamic part of this, but these molecules are really flexible. So you imagine these two arms, they can flop around. And so the flexibility is a key feature of these types of molecules too that will lead to some of their biological implications. What's really interesting to think about, and for a long time people would argue, like, there's so much heterogeneity. Cells are just sticking sugars on the cell just to have something there as a barrier, something to protect cells from whatever the environment might have in store. But my argument against that, and something we are still coming up with examples for why that's not true week after week, is that nothing in biology comes for free. There are reasons that we have genomic programs to make proteins that do things. 4% of our genome, or a few hundred to about 1,000 genes, encode for enzymes that regulate this really complex process of glycosylating things. So there's a lot of energy input and genomic material that's dedicated to this process. And so we want to understand, how is this regulated? We come back to this and we think about well, what would be the purpose of being able to modify proteins with sugars? Why would biology spend the effort to do this? And so what I want to impress upon you is that the genome is rigid, necessarily rigid. If you have a non-rigid genome, if your genome is mutating often, that's very bad for you. That is where things like cancer arise from. And we want the blueprint to stay the same. But that rigidity means that you can't adapt to your environment super well unless you have some way to have built-in flexibility to this system. Proteins are a much more flexible platform than the genome. But even on top of that, how could you dress up a protein to be the same sequence? You didn't have to change anything about the protein you made, but you could change how it acts based on what modifications you give it. And this is the biological implication of having sugars on protein, that versatility to adapt the function of molecules to whatever the environment is giving you. And so here's an example of the spike glycoprotein from SARS-CoV-2. This is the protein structure itself without any glycans on it. But what we know is it has multiple N-glycosylation sites, meaning they're linked through the nitrogens on the protein backbone. And if you were to pick just one glycan structure, so again, we're not even talking about the heterogeneity that could exist, but one glycan structure, and to draw in the space that those glycans occupy on the protein surface, that's the dark blue that you see in this image. Look how naked this protein looks without its glycosylation. Clearly, the glycans are going to drive a lot of how this protein interacts with its environment. These are just different images where this is basically, I talked about how floppy these glycans are. So this image is showing you the space that glycans could occupy averaged over an amount of time, whereas these would be images of just the glycan in a static moment. But you can see how this contributes to changing of the chemical space that a protein has by adding these sugars on there. Analogies that I've found useful when explaining this to lab mates, family, friends, one of them would be Mr. Potato Head, a classic, right? So you have the same potato, it's your protein, and here you can change what you do with that potato or what that potato represents in Mr. Potato Head by changing the decoration. So the protein would be Mr. Potato Head without anything, and then the glycans would be how you decorate that face. Another example that I use often are thinking about an actor. The person, the actor has not changed, but the role they play can be very much assumed or dictated based on how they dress up and the actions that they do. So Tom Hanks, been in so many different types of roles, but if you were to look at Tom Hanks here, clearly a Mr. Rogers impersonation, versus here, if I had to ask anyone, what is the person that's dressed like that doing? Like, oh, they're in war. Or here, be like, this is train conductor, maybe you have some similarities, and this is where maybe some decorations have related functions where others are completely different. But this gives us insight into the situation that Tom Hanks is in here is very different than the situation Tom Hanks is in there, and the function Tom Hanks might be doing at any given moment, depending on how he's dressed up. This one I showed this morning, but at the end of the day, something I think it's important to think about is we think about extracellular collections of these biomolecules. Proteins without their glycans is a poor representation of what they're doing. And so this would be the world that I think in, and it's a fun world, more decorated, it gives more flair when we think about glycoproteins. And so now I want to give you a few examples. This would be the take-home message of like, why should you care about all of this? I'll give you things you've already interacted with that you didn't know were because of glycosylation. But I'll mention why you may not know it is because, well, why are glycans so often ignored? I even mentioned this in the beginning where we have the mountain there and it's staring us in the face if we know it's there, but we totally miss it if we're not looking in the right way. Well, if you think about a lot of tools we use to measure biomolecules, they're very good at measuring static things. Even You may have heard of the structural biology revolution that's happened in the past 10 years with tools like cryo-EM, cryo-electron microscopy. It was really good at capturing structures of proteins, but those proteins are static. They're frozen in ice. Glycans, super floppy, very bad at being static. And so when people look to study proteins, almost all of the structures we have in what we call the protein database, protein data bank, are non-glycosylated proteins, even for proteins we know that should be glycosylated, because we can get a really good picture of a static person. We'd have to do these kind of quote-unquote long exposures when we try and take these images of these biomolecules. But the glycans, they're moving around. They're much more dynamic. And so when we take an image, this is the only thing we can really focus on, and we see blurs of what could be there. So this gets back to what are the tools we're using to study these biomolecules, which we won't, again, talk so much about tonight. But this comes back to the accompaniment of a biological question and a need, and the tools you're using to address that question and need. When it comes to the types of things glycans are affecting at the cell surface, there's a whole host of things, and this is far from an exhaustive list. But I wanted to show you here, this is the image of a cell surface that has glycoproteins and how things like ligands bind to other proteins at the cell surface, cell adhesion, so many things that the details of this, I hope I don't go too far down the rabbit hole, but it's to show you that it's affecting a lot. And one that I want to talk the most about is the immune system. So here, this would be an example of an immune cell that has different receptors on it. It turns out we have professional receptors on our immune cells for glycosylated proteins. Again, yet another argument that this isn't biological noise, but an actively regulated process. And so we're going to talk about what some of those look like. And again, we could teach an entire year's worth about the immune system, and the rabbit hole goes very deep there very quickly. So we won't dive into all of the intricacies there, but just to remind you, some of the language of the immune system would be things like pathogens that are now an insult on our healthy system. And we have things like monocytes and macrophages, neutrophils, B cells, T cells. Again, the details here I don't want you to get lost in, but just, these are some, if you've heard of this space before, this is the exact space where glycans can really matter. And so the first one that I want to mention are blood types. And if you didn't know this, your blood type is defined by enzymes that put different sugars on your blood cells. And so hopefully everyone here knows their blood type. It's an important thing to know. But for example, I'm A blood type. This means that I have an enzyme that people that are O blood type don't have. That enzyme takes this sugar molecule that's on the end of glycan. So the glycan could be very long, but at the very end of it, we have this signature. And I have an enzyme that puts on this GalNAc, so that yellow circle. And that now is the antigen that my body can recognize. But if my blood, A-type blood, went into someone with B-type blood, their body would see that and say, that's foreign, kill that. And that's why you have bad reactions if you don't have the right blood in your transfusion. And conversely, people that have B-type blood have an enzyme that's very similar, but here's the cool thing about glycans is the structure of these glycans matter because that enzyme puts on a galactose. So either a GalNAc or a galactose. The difference is just one acetyl group on this sugar. And all of a sudden, it's an entirely different molecule that your immune system recognizes. And these two are both different than if you don't have that sugar there. So this is just one area where glycans really matter. And it's not something that we teach as much as we should. And so this is kind of an intro in some of the ideas that we want to think about. Another one is viruses. And this is a little busier of a slide than I maybe should have put up, but what I want to emphasize to you is that they're the ends of glycans here. So these are structures, again, of these sugars. And these are called sialic acids. We've kind of mentioned them before. They're the purple diamonds. And so this cartoon here is showing the one, if that linkage were there, and this cartoon is showing if this linkage were here. And what the difference here is showing this, there's an alpha 2,6, which basically is where is it linked, or an alpha 2,3, which is showing that it's linked at that oxygen. That difference is the difference between influenza that affects birds versus that affects humans. You've heard of the bird flu. And why do we worry that bird flu could have a zoonotic jump into humans? Well, if the virus evolves to change which linkage it's using for that sialic acid, all of a sudden our receptors can recognize the virus, it binds to our cells and can infect us. So this is really, again, a nuanced difference in these two glycans, but it makes all of the difference in the biological implications of what happens. So just to kind of show you the life cycle of what a virus would look like here, we have viruses that are interacting with glycoproteins throughout their residence time at the cell surface. And basically we have receptors that can recognize and bind the glycans that are on the virus, and that's where that switch really matters. And so if we then all of a sudden, the virus has glycans that our receptors recognize, we start internalizing it and we get infected with the flu. What's cool, we've known about this for a while. Has anyone here heard of Tamiflu before? Tamiflu interrupts this sialic acid interaction. So basically it is an inhibitor for the enzyme that affects sialic acids on the virus cells. So it's going in and inhibiting the ability to make the enzymes interact with that sialic acid so that the virus then can't infect you. So again, things that you've interacted with before that you certainly have heard of the flu that now are being driven by glycans that you may not have recognized. A few other examples, viruses are a huge space where glycans matter. These would be examples of MERS and SARS, so different coronaviruses. And you can see here the colors are different glycans, and they differ by their glycan type. We talked about heterogeneity, that not all the glycan structures are the same. And so what's interesting here, the gray is a protein structure. So here we're looking top down on top of a protein, and here is that protein if we were looking at it, from the side. And so you can see different parts of the protein have different glycan types, which are color-coded here. And that absolutely affects how these viruses interact with all different cell types. And even lung cells versus skin cells, like what's the difference in how these viruses infect different cell types all depends on receptors that are interacting with the glycan protein combination of these virus proteins. Last thing I'll say is that so many viruses have glycosylation on them, and we've looked at even ones that have the density of glycosylation. We can measure how much of that protein surface area is covered by glycans versus not, and basically how much we might have different infectivity issues with these viruses. So glycans are playing a huge role in this. Another example that I want to give, which again just kind of highlighting reasons you might want to think or know about glycobiology, would be organ transplantation. This is a really exciting and interesting field. How do we, as a science community, get better at using resources that we already have at our disposal to improve human life? Well, a lot of different organisms have similar size organs to us, and pigs actually are one that have similar size organs and a number of heart, liver, pancreas, kidney. You can see these are like the general size comparisons between human and pig. And so there's all sorts of other things to address here, but a main limitation are the differences in glycans between pigs and humans. And so let me give you a few examples. I'll mention that people have looked at all sorts of different types of animals for this. And one thing we really don't want is zoonotic risk, and that comes back to the idea of what we're looking at with bird flu. There are other infections that affect other animals that don't affect humans. And we don't want to introduce the ability into humans to be infected by these other diseases. Turns out pigs are a really good match for us. We have really high overlap in the types of diseases that we already share together, so it's not as big of an issue. But it turns out the immune response to glycans that pigs have is the main limitation for why we can't quite use pig hearts in heart transplantations that we would need for patients. And there are 3 different glycans that contribute to this. The first one I want to point out is called alpha-gal. This is where we have 2 of these yellow circles, galactoses, linked by an alpha linkage at the 1,3. We didn't draw the structure here, but basically they're linked in the particular orientation. You may have heard of alpha-gal before because ticks can actually cause this to be an issue as well, where if you're bitten by a tick that introduces this antigen into your bloodstream and your body mounts an immune response to it, now you have antibodies to alpha-gal, and the next time you eat any meat, like from a cow or any other animal that has alpha-gal, you have a severe allergic reaction. So you may have heard of alpha-gal syndrome, and this is the exact glycan that's contributing to that. This also is a huge problem if you're trying to transplant an organ, because your body says, that's not human, those cells shouldn't exist in my body, please destroy them. Another one would be the sialic acid that I've mentioned before. Humans do not have the ability to process this purple diamond any further. Other mammals have one additional enzyme that we've effectively lost. And they can make this nu5AC into a nu5GC. And again, the details of this don't matter, but you can see that there's basically one additional chemical group that's attached. This also, your body sees that's foreign antigen that should not be here, attack and kill cells with that. One hypothesis is also that inflammation that you get from eating things like red meat is coming from reactions to glycans in your diet that are this way. More of a hypothesis than a proven point yet, but just again, trying to connect things you guys may have heard of before. Another one would be in this glycan that's on lipids that we just simply don't make as humans, where this is what we normally have, and pigs have this enzyme that then make this. How do we address something like this? Well, what's really cool is if we understand that 4% of the genome that's controlling how glycans get made, we can then edit this in the organism. So what we can do is we can knock out enzymes that are contributing to these glycan features. And so we can make transgenic pigs that don't make, so go back a slide, any of these. All of a sudden, we now could very well have a heart from a pig that could be very functional in a human without any immune rejection. This has been tested. It's still very much in the early phases. I believe people have lived for months with a pig heart now. Basically folks that are out of any other luck but to try an option like this and have had minimal success to date. But it's a cool space, and you're interested in like tissue engineering and the bioengineering world, very much a cool space where glycans matter. And the last one we touched on this morning that I'll expand slightly on, and then we'll call it a day, and just hopefully by that point you all convinced that glycobiology is a very cool space to be studying. This one has to do with the immune system and cancer. And the idea here would be that we have what we call immune checkpoints. And so you can imagine that your immune cells have receptors on them that are constantly feeling in the dark, and they're feeling cells that are you, that should be there. And when they see self, there's a mechanism inside them that puts the brakes on and says, that's us, don't kill it. Well, it turns out cancers have evolved to upregulate proteins that bind those same receptors. And so the cancer is tricking the immune system. It's saying, I should be here. And the immune system says, we believe you, we won't kill you. And so one of the protein interactions that is involved with this is called PDL1, programmed death ligand 1. And so basically when cancers upregulate this protein, this would be a cancer cell here, now it's on their cell surface, your immune cells have a receptor for that protein called PD1. The internal machinery here is a whole cascade of PTMs, phosphorylation in this case, totally different than what we're talking about today, but another cool space to think about. But that cascade says, don't kill, put the brakes on. And this has been an entire success story, limited but still successful, of what we call immunotherapy in cancer. And so this has been a huge advantage for a lot of blood cancers. So think of leukemias, myelomas, things like that, and also in melanoma. But there's still a struggle in solid tumors to be able to use this axis effectively. And so other things that cancer cells do, they don't don't just upregulate proteins like this. And so cancer cells are trying to basically hide from this interaction. So we have immune cells in green and our cancer cells in pink. And the immune cells are effectively just going in and let's call it tasting. They're saying like, does this seem good? Does this seem bad? And what cancer cells are trying to do is trick the immune cells into saying that we're good. And so we come back here. This is interaction where because this protein is there, the immune cell says check mark. And it turns out cancers have learned that immune cells also check for sugars, specifically for, again, these pink diamonds called sialic acids. So healthy cells have some amount of that, but they also don't have any other antigens that might give a warning sign to the immune system. So they don't need to have a total abundance of this. They just have some of that. And immune cells see that and they say, healthy cell, don't kill it. Cancer cells have evolved to upregulate proteins that are really densely modified with these glycans. So cancers have other things going wrong that might alert the immune system. But they upregulate proteins like this that will mask that, and they're basically cloaking themselves, a good word, we were talking earlier with Eric about this, that we now can hide cancers from the immune system. So what this looks like is we actually have receptors. In this case, we're looking at ones called Siglecs. And the name behind this is not so important, but the thing to key in on is they have the exact same internal machinery as the PD-1 receptor does too. So as soon as these proteins bind these sialylated glycoproteins on cell surfaces, they put the brakes on as well. What we're starting to appreciate is that it's not just any receptor that recognizes any glycoprotein, but now specific receptors are recognizing certain protein sequences with exact patterns of specific glycans. Which ones? Great question. We don't know. And this is a cool space that we're actively trying to figure out. What are those molecules and what are the surfaces that look like with the Siglec binding a glycoprotein? What is the surface that matters? And how do we disrupt that selectively in cancer without touching Siglecs that are really useful for normal immune function? So a few thoughts that have come out if you're thinking about how are people doing this right now. This is a cool space from Carolyn Bertozzi's lab. So again, biased in sharing this example because it's from the lab I worked in. Their goal was to basically try and target cancers that have this upregulated sialic acid. And here's that same kind of picture we're looking at shown in a different way. This is the immune cell, this is the Siglec, and this is the cancer cell that has upregulated all of these glycoproteins. And the treatment is to basically target with an antibody, basically a homing mechanism, that brings an enzyme to the cancer cell. So this little Pac-Man is an enzyme, this is the antibody. And by bringing that enzyme to the cancer cell, they want to basically take a lawnmower and cut off all of the purple diamonds, all the sialic acids. It's kind of like a bazooka, right? It's taking everything off of the cancer cell, but it's a cool space to be thinking about. And so this is now in phase 1, I believe, trials. People are trying to see if this can improve cancer immunotherapy. One thing my group is interested in is how do we get a bit more refined with what we target? So this comes back to the mass spec world, again, of how do we study what these specific molecules are. So I think what we have focused on right now is the functions of glycans, but the last thing I want to leave you with is this idea that I just mentioned, that it's specific proteins with specific glycans that matter in these interactions, and we call this idea the glycocode. And this is the combination of that glycan at that specific location on a protein. That is the unit of biological information being conveyed. And this is what is analytically challenging to capture, but where we're working on technology to get better at studying this in really complex systems where there's hundreds of thousands of these types of molecules around. This is, I'll just give the basic examples. I would point out that the way to target Siglecs, I gave you that one example. This is another depiction of the same idea where an antibody is bringing an enzyme, represented now by scissors. But people are thinking about this space in all sorts of ways. So it's a really active space in cancer immunotherapy, and we won't need to go through all of these examples, especially for time. but that this is, if you're interested in cancer and glycobiology, a fascinating world that's actively being investigated right now. I'll also mention that the idea of Siglecs, there's a whole host of them. So it's not like it's just one receptor. There's so much we need to study. This, to me, is what I see this slide, and I love it because it's about opportunity. There's so much we don't understand that we want to be able to study. And so one that my group is focusing in on is called Siglec-10, and it's been basically studied in a couple cancers. And if you haven't seen these before, they're called Kaplan-Meier curves, and they're, to me, very devastating curves to look at because they're showing survival rates. And these are basically thousands of patients that have been compiled, and they're studying the transcriptome of samples from these patients. And in this case, we'd say that these are 5,000 ovarian cancer patients, and we can look at the difference between patients that had a high expression of this protein on the cancer cell called CD24, or a low expression. And you can see the difference in survival rates if you had high versus low. Again, devastating graphs to look at, but very useful for showing us important targets to go after. And so here, breast cancer is another one, not just ovarian cancer. And what's interesting is that we see CD24 is exclusively expressed. These are basically a map and showing you where CD24 is and some other different types of proteins. That CD24 is on tumor cells, and Siglec that we're interested in is right here. It's on what we call tumor-associated macrophages. So these are the immune cells that are in the tumor microenvironment. So this interaction is happening and causing cancer cells to keep living. So we're interested in studying this. I'll skip this for detail, but just to show you that this is the interaction we want to understand, and how does that look like in an experiment in our lab? Well, a question would be, if you had ovarian cancer or breast cancer or even lung cancers where we've seen this be a challenge, be like, what are the glycoproteins that are interacting? What are the different modifications on CD24 that drive the interaction between the cancer cell and the immune cell? And so we're trying to use mass spectrometry to say, can we map all of the different glycoforms? Can we fingerprint what the glycoproteome looks like in ovarian cancer, in breast cancer, in lung cancer, et cetera? Do those proteins look the same? That would be interesting. Or do they look very different in their modification states? Also interesting and useful when designing therapies. Again, I mentioned the interface then of when Siglecs bind these proteins, how do we map the topology of that interaction? Then also, when we look inside of cells, can we measure these signaling cascades as this interaction happens? So when we think about the technology we talked about this morning, if you were at that talk, in the mass spec world, this is the other side. This is the biological questions that we center in the middle, like the target of our methods. I'll end with this idea to say, we led the talk out with M&Ms, and hopefully, now when you see M&Ms, this is what I tell you, when I see M&Ms, now I see this, sugar coating on the cell surface. And so, if nothing else, hope you've learned a little bit about glycans and how they're important to things you already have heard about, but maybe didn't know is because of sugars. I'll also mention, he stands in the audience, but you have someone here that studies sugars too. We talked a lot about glycoproteins, where proteins and glycans tend to be equal in mass. There's also cool molecules called proteoglycans, where the glycans themselves are a huge component of mass. So we have people here working on this if you're interested in that space too. And this is, again, to emphasize the idea of crosstalk between methods. If you can't study molecules, if you're blind to them, then all of this doesn't matter, right? We have to be able to capture this. So this gets back to the idea of the chemistry side of this, of how do you build tools to manipulate and leverage the chemistry of sugars and proteins to be able to study these in the systems we care about. This is a collection of projects in my group that we don't need to go through, but just to show it's a really fun and wild world for hard molecules like glycoproteins. I mentioned I'd end with this slide, so we're here now at the end to say thank you to you all for your attention. A huge thank you to the university and to Dr. Pribble for all the hosting and invitation to be here. Thank you to the Ford family. And thank you to the people in my group who are leading the cool science. It's a fun space to be thinking about and fun to interact with teams of people. Science is about teams of people. I'm very thankful to have these people on my team, and thankful for you all for being here this evening and listening to this talk. I'd be happy to take any questions.
we do have time for questions. I ask that you raise your hand and we get the mic as we are recording, and we want to make sure everyone can hear.
So my dad has metastatic melanoma, so this is actually really cool to hear about, and he has taken some immunotherapy treatments. I was wondering, do you guys do a lot of like the breast cancer and the ovarian cancer, or do you do like stuff with melanoma?
It's a great question. So right now, most of the models we're using are quite removed from like an actual in vivo patient population, anything like that. We're using cell line models, which are very useful for kind of these baseline technology developments. Eventually, we want to get into where we could use these methods to actually study samples from patients and understand how immune therapy is progressing or changing the glycoproteome. Our main focus right now has been breast cancer and ovarian cancer because there are really good models with cell lines for metastatic, like basically progression in that way. Actually had great conversations earlier today about how to expand some of the models we're looking at. But I think that is what is interesting about being in a method development lab, is that the goal would be all these tools we're looking to study glycoproteins could be broadly applicable. It's just about being able to say, like, this system matters, and we want to apply it to that. So right now, and there's nothing saying that the mass spec couldn't be used for fill in the blank. It doesn't have to be cancer. It could be other disease states that we want to study. But we are trying to focus on metastasis and a few cancer models just to start with.
Thank you.
Yeah.
Thank you so much. Both of your lectures are just fabulous.
Thank you.
Really great. This may be, in response to your answer to the last question, this may be a little downstream from you, but I'm sitting here thinking about the supply chain of monosaccharides needed by cancer cells in order to express the volume of sugars on the surfaces that are needed in order to do this heavily evolving landscape of fooling immune cells. And I'm thinking about what we know about in many types of cancers. One of the things you can do is move to a ketogenic diet so that you're not ingesting any forms of carbohydrates, simpler or complex. And that this can slow down even, for instance, the most aggressive brain cancers for a few years. In the end, you lose, but you buy yourself some time. Is there anything to that? Is the total amount of monosaccharide volume needed related to the ingestion at the human body level? Is it related in any way to what you're doing?
So I will say related, absolutely, but how directly is challenging, because metabolism is so multifaceted. The area that I've seen of most interest, at least in the work we're doing, is something called the hexosamine biosynthetic pathway. And it's basically how different glucose and sugars get shunted into that GLICNAC, the blue square, as one of the precursors. And there's a lot of inputs to that pathway. And so being far from a metabolism expert, I couldn't say like ketogenic diet for sure can point to that being a pathway of interest here. But what I can say is we've done some cool collaborations recently with people that are mitochondrial experts. And they mess with mitochondrial metabolism, and we see pretty dramatic changes in cell surface glycosylation. Mitochondria have, in theory, nothing to do with the cell surface. And so it's all intertwined, right? Like these are complex networks of how biomolecules get shuttled around. But I think there's a lot to still explore there. What is really cool, so basically what Carolyn Bertozzi got one of the Nobel Prize for is figuring out a way to feed sugars to cells that have unnatural side chains on them. And she could feed cells the sugars, they would append them to the cell surface, and she could use that as a handle to then study the sugars. That process is called bioorthogonal chemistry. But what that tells me is that there's a lot of inputs that basically cells could take, even molecules we don't expect them to be able to use, that they could be then converting into precursors for these sugars. So I think that there's a whole world of like flux metabolomics. So people using tools like mass spec to study how different sugars get shunted down these pathways. And that's, again, not an expertise I have, but a cool space where as we get better at studying glycans, it's still very hard to even study the glycans in the first place. as the tools keep maturing, we can then start saying in really complex experimental design, like how do we say, yes, metabolism altered in this specific way, here's the glycan features we see, maybe we can connect the dots that way.
I'm going to take up all the air in the room if we're not careful. I was struck by the fact that phosphorus cannot be generated by the human body, but glycogens can. And you can live without ever consuming carbohydrates, but you cannot live without consuming phosphorus. And I just wonder if you have any thoughts on that, given that these are the two fundamental components of what you're talking about.
I think it boils back down to where sugars, there's so many molecules that use sugars, and what the precursor requirement looks like to build the glycan is quite minimal. You need something called the CMP sugar acid. And so those can be generated in a lot of ways, even from breaking down things like nucleic acid. So there's a lot of, because nucleic acids have a sugar backbone, right? And so there already are carbon sources that can be used by our metabolic pathways to make some of these precursors. But again, not metabolism expert by any means. But that, I mean, that would be my off-the-cuff thought to that question.
Other questions for the speaker today?
I probably have a more simpler question. I was just intrigued by what you were talking about, blood types, and how each person has a particular blood type. Is that genetic somehow? When your blood cells are formed, they're already ready for certain glycans that they're just, the glycans aren't there yet, but then they just show up because the cell has sort of the characteristic that work with certain glycans?
Absolutely. So the genetic component of this is like, do you express either enzyme to make A type, B type, or do you have both enzymes and make A, B, O type? And so that is genetic. It's encoded in the blueprint you have. So all that to say, you're already primed. You're born with the ability to make one of them or none of them that way. A cool space people are looking at is how do you convert any blood type that someone would donate into a universal O type. We basically are using enzymes to take the glycans off after someone's donated blood, and now it becomes O type, and everyone will be okay receiving O type, right, the universal donor, because it doesn't have that bonus sugar that you would recognize as an antigen if you didn't have that as a normal blood type for you. I don't know if that makes sense. But yeah, to answer your question, the enzyme that puts the sugar on, genetically encoded.
Please join me in thanking Dr. Riley for a wonderful presentation. I know I will not look at M&Ms the same. And I'm quite sure some of the folks here will not. A couple last things. Stay up here. I'd like to present you with your own copy of Chemical Magic, written by Leonard Ford and revised by Wynne Grunmeyer, one of his students and who became one of his colleagues. So please enjoy that. And because we are chemistry, a test tube pin to remember your time at Minnesota State. So thank you, Nick.
Thank you very much.
The lectureship just doesn't happen by itself. There are a number of people behind the scenes and in front of you that do work. Chris Kors, our administrative assistant, does a huge role that's always behind the scenes. She's not here tonight. She would be very embarrassed when I say her name, but she does a lot of work behind, so I thank her. I thank Dr. Swart, Dr. Vorlachek, Dr. Moen, who all help with a variety of the organization of this, and for you for showing up tonight. One last thing, out in the hallway we do have refreshments hosted by the Chemistry Club and Biochemistry Club. I wish you a great evening. Thank you, and I look forward to seeing you next year.